Hello everyone, welcome to the class. How are you all? So today we are going to discuss absorption of food. Yes, which is again an important part from the um, digestion there. And if we can see from the physiology or animal physiology, isn't it? So it is again absorption. It is interesting topic also, isn't it? यदि हम digestion पढ़े तो digestion सारा देखने में it becomes little bit boring, but yes, when we go with an absorption part, absorption is really very interesting. So today we will discuss about this, yeah. And yeah, we are going to discuss this topic in two parts. So this is some of the absorption we are going to discuss in today's class. So absorption of food, if I am talking, यदि मैं बात करती हूँ absorption of food की, we know that our food is completely absorbed, yes, in small intestine, isn't it? Most of it, yeah. If some part is remaining the water, and some of it we can say it happens in the large intestine, but maximum 90 to 95% of all the nutrients are absorbed in the small intestine only, yeah. Now this absorption, it takes place via, you can say it can take place with a simple diffusion, it can also occur through facilitated diffusion or through the active transport, है ना? किस तरह से transporter रहेंगे, which type of transporters are there, whether it is moving through transporter, whether it is moving as it is, it according to that we can say that the transport of the or the absorption of the materials or the nutrients takes place. Now before moving for the main topic of absorption, just a small information for you all that you all must be knowing, आप सभी को पता होगा कि there are two non-nutrient substances which are not nutrients, which are absorbed directly by our stomach. What are they? It is ethyl alcohol and aspirin. Okay, so what is it? Ethyl alcohol and aspirin. You can see here. These are the two non-nutrient substances which are absorbed directly by the stomach. Now, if I am talking about absorption, we know that in intestine there are villi, isn't it? The finger-like projections, villi, whose epithelial cells are called as enterocytes. And these villi are responsible for the absorption of the nutrients. Yes, it is present in the small intestine as well as in a large intestine. आप सभी को ये सारी चीज़ें पता है, जब हमने digestive system पढ़ा था in detail, we have seen all these things, isn't it? So the apical surface of the enterocyte, they have this microvilli, and this microvilli they help in an absorption. So first of all, we are going to discuss absorption of monosaccharides. Yes, आज हम पढ़ेंगे absorption of monosaccharides का how it happens. So we know that if I want to absorb a carbohydrate, किसी भी carbohydrate को absorb करना है तो वो monosaccharide के form में ही absorbed होगा. Yes, always remember that the carbohydrates are absorbed in a form of monosaccharides. Now monosaccharides, how the absorption will take place? It takes place through the facilitated diffusion as well as active diffusion. दोनों तरह से होता है. कौन सा मोनोसैकेराइड किस तरह से एब्जॉर्ब होगा, वी विल डिस्कस वन बाय वन. नाउ इफ आई एम टॉकिंग अबाउट एंटेरोसाइट, यस, यदि मैं एंटेरोसाइट सेल की बात करती हूँ तो ऑलवेज रिमेंबर दैट व्हाट मेम्ब्रेन ऑफ द एंटेरोसाइट व्हिच इज फेसिंग द ल्यूमेन, we called it as apical membrane or luminal membrane, है ना? मैं why I am saying because when we will go ahead, जैसे जैसे हम आगे बढ़ेंगे, काफ़ी बार there will be the words like it is absorbed from the luminal membrane or apical membrane and it is going out like that, isn't it? So luminal membrane या apical membrane कौन सी रहेगी? जो membrane है which is towards the lumen, which is facing towards the lumen, the which is having the brush border of the villi, isn't it? And the opposite, the basolateral part, yes, which is facing the blood of the capillaries. Got it? So this you should remember these two things. Now coming to the part again, if I am talking about the absorption of monosaccharides, students, on an apical membrane, yes, एंटेरोसाइट के अपाइकल मेम्ब्रेन में कुछ ट्रांसपोर्टर्स होते हैं विच आर रिस्पॉन्सिबल फॉर द एब्जॉर्प्शन ऑफ मोनोसैकेराइड्स. विच आर दैट ट्रांसपोर्टर? द ट्रांसपोर्टर विच इज कॉल्ड एज SGLT1. यस, और दूसरा ट्रांसपोर्टर है जिसको बोलते हैं GLUT5. ये दोनों ट्रांसपोर्टर विच आर दे आर प्रेजेंट एट एन अपाइकल मेम्ब्रेन. Yes, कहाँ पर है? They are present at apical membrane of a enterocyte. This both transporter they are responsible for absorbing the monosaccharide. SGLT1 ये एक symporter है. Yes, this is a symporter. Now I hope आपको पता है what is symporter. Yes, that means it will take two things together, isn't it? So this symporter or SGLT1, what is the full form? Sodium glucose, yes, transporter there, isn't it? Now students, this will take always the monosaccharide with sodium. That's why it is sodium glucose linked transporter, SGLT1. है ना? तो हमेशा ग्लूकोज के साथ साथ किसको अंदर लेगा? वो अपने साथ में सोडियम को भी लेकर आएगा. बोथ विल move in a same direction, that is a symporter. There are antiporter also, but I am talking about symporter. GLUT5, it is also a protein transporter जो कि मोनोसेकेराइड को ट्रांसपोर्ट करने में हेल्प करेगा. ये दोनों आपके ट्रांसपोर्टर कहाँ रहेंगे? दे आर प्रेजेंट ऑन अपाइकल मेम्ब्रेन. ओके, so now remember one thing, here it is written as monosaccharides enter into the cell through apical membrane via facilitate diffusion और एक्टिव डिफ्यूजन. एक्टिव डिफ्यूजन से कौन एब्जॉर्ब होता है? ग्लूकोज और गैलेक्टोज. whereas facilitate diffusion से the fructose is transported. Okay, so I will write here that the glucose and galactose, yes, it is absorbed. How it is absorbed? By active diffusion. So it is a secondary active process. Yes, and this is absorbed by secondary active process along with, I can say, sodium. Yes, and the transporter, it is your SGLT1. Yes, so SGLT1 किसका ट्रांसपोर्ट करता है? किसको एब्जॉर्ब करेगा? इट विल हेल्प टू एब्जॉर्ब व्हाट? सोडियम, सॉरी, ग्लूकोज एंड गैलेक्टोज. लेकिन इसके साथ साथ सोडियम भी सेल के अंदर जाएगा. जस्ट रिमेंबर दैट. एंड इफ आई एम टॉकिंग अबाउट फ्रुक्टोज, yes, now this fructose, how it is transported? The fructose, it is absorbed, yes, by facilitated diffusion. And it is done by your GLUT5. Again, I am repeating that both of them, they are present at an apical membrane of a cell. Okay, now there is one more which is called as your sodium potassium ATPase. Yes, sodium potassium ATPase is also there, which plays a very important role. Yes, it plays a very important role in absorption of monosaccharides. क्या रोल प्ले करता है? कैसे हम बोल रहे हैं कि it is playing an important role? Now listen, I'll explain you with the figure only. You can see here. Just a minute. Yeah, you can see this one. This is an enterocyte cell. Yes, ये एक enterocyte है. Now this is the luminal side and this is your submucosa. Yeah, so ये आपका जो है ये luminal membrane है और ये आपका हो जाएगा basolateral membrane. Got it? So आप देख सकते हो luminal membrane से SGLT1 क्या कर रहा है? It is transporting or it is helping to absorb glucose and galactose along with sodium. Yes, it is a symporter. So sodium और glucose galactose सेल के अंदर आ रहे हैं through SGLT1. इसी तरह से फ्रुक्टोज सेल के अंदर आ रहा है, yes, through the transporter which is called as a GLUT5, glucose transporter 5. Got it? यहाँ तक समझ में आया? जब मैंने बताया कि सोडियम पोटेशियम एटीपेज है, दैट इज ऑल्सो प्लेइंग अ वेरी इंपॉर्टेंट रोल. कहाँ पर है? वेर इज दिस सोडियम पोटेशियम एटीपेज? हियर यू कैन सी. So this sodium potassium ATPase, it is present at the basolateral membrane. Yes, it is at the basolateral side. This is the basolateral side. Got it? So ये कैसे help करता है? Come on now, listen it carefully. Okay. Now as the glucose, first of all, as the glucose is absorbed, yes, from this luminal or apical membrane with the help of SGLT1, what happens? Along with the glucose, sodium is also entering. ग्लूकोज के साथ साथ सोडियम भी सेल के अंदर एंटर होगा. So जैसे जैसे ग्लूकोज का एब्जॉर्प्शन ज़्यादा होगा, as the glucose is absorbed more and more sodium will also enter the cell. Yeah. So when the glucose concentration is more, because absorption of glucose is done, with that the concentration of sodium also increases inside the cell. एंटेरोसाइट सेल के अंदर सोडियम का कॉन्सेंट्रेशन भी बढ़ जाएगा. अब सोडियम का कॉन्सेंट्रेशन बढ़ने से क्या हो जाएगा स्टूडेंट्स? इफ देयर विल बी मोर सोडियम, then this SGLT1, it will not work properly. Got it? Why? Because the concentration gradient is disturbed. Outside inside का जो gradient रहता है, that will be disturbed, isn't it? So it is very necessary to maintain the concentration gradient of sodium. Clear? So if SGLT will not work, then there will be no absorption of glucose again. So that will be a problem. So for that, when the sodium concentration inside the cell increases, this sodium is taken out of the cell, yes, with the help of the sodium potassium ATPase. Yes, so this sodium potassium ATPase always remain active to pump out the sodium into the interstitial fluid. Yeah, so जो सोडियम का कॉन्सेंट्रेशन बढ़ रहा है, वो सोडियम एक्स्ट्रा का सोडियम बाहर निकाल देगा, ये सोडियम पोटेशियम एटीपेज है ना. पोटेशियम अंदर आएगा इन exchange of the sodium, and sodium it will go out and in the interstitial fluid. तो अब concentration gradient maintain हो रहा है. अब जैसे जैसे सोडियम बाहर जाएगा तो सोडियम का concentration सेल के अंदर कम हो जाएगा. Now the sodium concentration in the cell will become less. So maintain this sodium concentration gradient. What happens to sodium ion? More and more SGLT, it will take the sodium inside. और जैसे ही सोडियम को अंदर लेगा, सोडियम के साथ साथ आपका ग्लूकोज और गैलेक्टोज भी एब्जॉर्ब होगा. आई होप दिस इज क्लियर. सो दिस sodium potassium ATPase, it is maintaining the sodium concentration gradient between the lumen and the cell. I hope this is clear. है ना. जब सोडियम का कॉन्सेंट्रेशन बढ़ गया, सोडियम को बाहर निकाल दिया एटीपेज ने. जैसे सोडियम का कॉन्सेंट्रेशन अब कम हो गया तो क्या हो गया, आपका SGLT1 एक्टिव हो जाएगा एंड इट विल मेक द सोडियम फ्रॉम आउटसाइड टू bring it inside, and with that sodium who will also enter, because sodium will not enter alone, it is your symporter. So it will, if we want sodium inside the cell, with that it should combine with the glucose और galactose. तो इस तरह से glucose galactose का absorption होता है SGLT1 से, and in that the sodium potassium ATPase also plays an important role. I hope this is clear. Okay. About the fructose, there is no tension, है ना. Fructose is absorbed through GLUT5 directly, students. There will be no ATPase required or like that. It is by, and it is not an active transport. It will be from the facilitated diffusion. I hope this is clear. Yeah. Now once this fructose, glucose, galactose all has entered inside the cell, absorb हो गए. आफ्टर एब्जॉर्प्शन क्या होना चाहिए, वो सेल में से ये सारी चीजें ब्लड के थ्रू, इट विल एंटर द ब्लड, इजंट इट. सो वो बाहर कैसे निकलेंगी, दे ऑल विल डिफ्यूज आउट थ्रू द सेम ट्रांसपोर्टर, दैट इज GLUT2. So glucose, galactose and fructose diffuses into the blood from the basolateral membrane by a transporter which is called as a GLUT2. So how many transporter you have to remember? आपको 3 transporter याद रखने हैं monosaccharides या फिर carbohydrates के absorption में. One is your SGLT1, another is your GLUT5 and the third one is your GLUT2. GLUT2 से सारे diffuse out होंगे from the cell, and this both transporter, it will absorb the glucose, galactose and fructose inside the cell. Yes, it will help to absorb and it will help to diffuse out. यहाँ तक समझ में आया? I think now it is very much understood. So we can write, और इसको sum up कैसे कर सकते हैं? We can sum up like that. If it is a sodium, yes, sodium हमेशा आएगा along with glucose as well as galactose, yes, with the help of SGLT1. और किस तरह का ट्रांसपोर्ट होगा? ये होगा आपका सेकेंडरी एक्टिव ट्रांसपोर्ट. सेकेंडरी एक्टिव ट्रांसपोर्ट. Yes, it enters the enterocyte. Enterocyte में आ गया, and from the enterocyte it goes out with GLUT2. Yes, it will be again facilitated diffusion. ये facilitated diffusion हो जाएगा. Yes, it diffuses out in blood. I hope this is clear. यहाँ तक समझ में आया? यदि मैं बात करूँ सिर्फ fructose की, so fructose, it will be taken inside or it will be entered the enterocyte through GLUT5. This will be facilitated diffusion. And again, with the facilitated diffusion only, it will go out into the blood. And here there is a role of GLUT2. Clear, students? So you have to remember all these things. So this was about the absorption of monosaccharides. Now remember, the rate of absorption of fructose will be nearly half to that of the rate of absorption of glucose. Glucose का absorption ज़्यादा होता है fructose से. Got it? Now after the monosaccharides, the next it will be, okay, we will just take it out also. So what is given here? Hence the sodium potassium pump always remains active. I told you, मैंने आपको जैसे बताया, and in that way it is maintaining what? The concentration gradient of sodium between the lumen and the cell, isn't it? And similarly, we can say here that this, so glucose and fructose as well as galactose, they all will be diffused out, or they will be taken out of the cell by facilitated diffusion, which will be mediated by GLUT2. And where is this GLUT2 present? GLUT2 कहाँ पर है? It is present at a basolateral surface which is facing the interstitial fluid. Interstitial fluid में चला जाएगा वहाँ से. Got it? So we can say the energy for uphill transport of glucose, glucose को अंदर लाने के लिए जो energy है, yes, it is coming from the downhill transport of sodium along the electrochemical gradient, है ना? सोडियम का concentration कम होगा तभी तो SGLT1 काम करेगा और ग्लूकोज का absorption होगा. I hope this is clear. Yeah, here we can write here interstitial fluid. Now the next is the absorption of amino acids and peptides. Now what is there in the absorption of amino acids and peptides? Students, we know that 95 to 98% of proteins that are present in small intestine, they are digested and they are absorbed also there only, है ना? इंटेस्टाइन में कुछ एंजाइम्स होते हैं जिसको हम बोलते हैं प्रोटियोलिटिक एंजाइम्स. ये एंजाइम्स प्रोटीन्स को ब्रेक डाउन करेंगे. इट विल स्प्लिट द लार्जर पॉलीपेप्टाइड्स इनटू डाइपेप्टाइड्स, ट्राइपेप्टाइड्स एंड अमाइनो एसिड्स, है ना? सो कुछ अमाइनो एसिड्स में स्प्लिट हो जाएगा डायरेक्टली, कुछ डाइपेप्टाइड में रहेगा, कोई ट्राइपेप्टाइड में रहेगा. इन दैट वे दे विल बी स्प्लिट अप. नाउ if the proteins are converted into amino acids. Just remember, if the proteins are converted directly into amino acids, यदि amino acids में उनका conversion हो गया तो क्या होगा, ये amino acid सोडियम के साथ कपल करेगा, सोडियम के साथ मिल जाएगा, and it will be co-transported or it will be absorbed into the cell, yes, by active transport process. कब होगा, if the protein is converted into amino acid, but if it remains in a di or tripeptide form, okay, then special transporter is required, and that is called as a peptide-1, that is peptide transporter 1. क्लियर. तो ये जो पेप्टाइड ट्रांसपोर्टर के थ्रू जब डाई और ट्राई पेप्टाइड एब्जॉर्ब हो गए, दे विल नीड proton. Yes, so they will be coupled with a proton. So what I told you, if it is an amino acid, it will be co-transported along with sodium. If it is in a form of a di or tripeptide, it will be co-transported with proton by a transporter called as a peptide-1. Is this much clear? You can see here. You can see this figure. Here this is a protein, students. With the help of proteolytic enzyme, यदि मैं बात करूँ pepsin, yes, or pancreatic peptidases, isn't it? So this will be converted into oligopeptides as well as amino acids. यदि वो amino acid में convert हो गया है, so this amino acids along with sodium, students, ये देखो ये सारे transporter लगे हैं, they all are present at a luminal membrane, isn't it? So this will transport it along with the sodium. कैसे होगा students? Now this sodium, it will move downwards, yes, through the electrochemical gradient to the interior of cell. सेल के अंदर जाएगा through electrochemical gradient, और जैसे ही वो अंदर जाएगा वो अपने साथ साथ it will pull the amino acids along with it, and it will take the amino acid inside the cell. तो इसको हम क्या बोलेंगे, दिस इज अगेन अ को ट्रांसपोर्ट, और वी कैन से सेकेंडरी एक्टिव ट्रांसपोर्ट. गॉट इट. नाउ इफ इट इज प्रेजेंट, दिस प्रोटीन इज converted into small oligopeptides, or it is in a form of a dipeptide or tripeptide, तो वो सोडियम के साथ नहीं जाएगा, तो उसके लिए अलग ट्रांसपोर्टर लगेगा जिसको हम बोलते हैं पेप्टी-1. यस, जिसको हम बोलते हैं पेप्टी-1. यदि ये ट्रांसपोर्टर है तो ये किसके साथ ऑलिगोपेप्टाइड्स को लेकर जाएगा, इट विल टेक अलोंग विथ प्रोटॉन्स. Yes, similarly, as a proton will go with that, this peptidase, it will also enter into the cell. Peptidase भी अंदर आ गया, amino acid भी अंदर आ गया. अब students, once it comes inside the cell, this peptidase, it will be broken down again into amino acids. ये amino acid में broken down हो गया या convert हो गया. That is necessary. Yes, once they are converted into this amino acid, now this amino acid with the amino acid transporter, it will diffuse out into the interstitial fluid. I hope this is clear. यहाँ तक समझ में आया? Yes, it will go out into the interstitial fluid with the amino acid transporter. So, if the protein in a form of di or tripeptide also it is absorbed, it can be absorbed. Absorb हो जाएगा, लेकिन absorb होने के बाद में it has to again converted or broken down into amino acid. Then only from the cell it will be taken out into the interstitial fluid. Clear? So what we have seen here, this was absorption of proteins there. So what we have seen in a simple way, I can write here, if the protein is in a form of amino acid, यदि वो amino acid के form है, so it will combine or it will be co-transported along with the sodium. Co-transport. Yes, or we can say it is secondary active transport. And it will enter where? It will enter the enterocyte. That means it is absorbed, है ना? Absorbed हो रहा है अंदर. and from the enterocyte it will go out. If it is an amino acid diffuses into interstitial fluid by amino acid transporter. Got it? Now, यदि it is in a form of di or tripeptide, if it is in a form of di or tripeptide, this will along with a proton, yes, they will combine with the proton, and it will take a help of a PEP-T1 as a transporter, yes, and they will also enter where? They will enter into the enterocyte. Then in the enterocyte, this peptides will be converted to amino acid, and then this amino acid again diffuses into the interstitial fluid by same way amino acid transporter. Got it? ये समझ में आ गया? And this will be by the facilitated diffusion. Clear? So this was absorption of amino proteins. Now the next is your absorption of lipids and fatty acids. Again, this is also very interesting, students. Lipid and fatty acids, we know that the lipids, the fatty acids can be long chain fatty acid, it can be short chain fatty acid. यदि short chain fatty acids है, that means 12 कार्बन से छोटे, yes, less than 12 carbon, they will be absorbed directly through simple diffusion. सिंपल डिफ्यूजन से वो डायरेक्टली एब्जॉर्ब हो जाएंगे यदि शॉर्ट चेन फैटी एसिड्स है. बट इफ इट इज अ लॉन्ग चेन फैटी एसिड्स, yes, or it is in a form of a mono or diacyl glycerol, then they cannot be absorbed by simple diffusion. वो simple diffusion से absorb नहीं होंगे. So वो क्या होगा? पहले they will form a micelle, yes, with the bile salts. जो bile salts रहेंगे उसके साथ में they will form a micelle. Now you all know what is micelle, isn't it? Now this micelle, it will fuse and enter into the enterocyte. यानी वो किसके साथ में enter होंगे? They will form the micelle and then they will enter into the enterocyte. आप देख सकते हो fatty acids and monoglycerides are there, है ना? These are in a form of a long chain fatty acids. We are talking about a long chain fatty acid. तो ये जो long chain fatty acids है, ये पहले they will be converted as, they will form as an emulsion, yes, और बाद में they will form the micelle there, yes, and it will enter in the form of that, it will enter from the brush border जो है आपका intestine का, villi का, वहाँ से अंदर आ गए. Yes, after that what happened? As this micelle is internalized, yes, once they will come inside, अंदर आने के बाद में क्या होगा? ये जो fatty acids थे, yes, ये ने जो मोनो और या फिर diacyl glycerides और fatty acids थे, they had formed micelle. Now they will be get separated, yes, and again they will be resynthesized into triglycerides. फिर से वो triglycerides में resynthesized हो जाएंगे. और जो मिसली जो फॉर्म हुआ था दैट विल गो आउट, वो बाहर चला जाएगा फिर से. नाउ दिस triglyceride students, they will be packed along with the cholesterol, yes, into a protein called as a chylomicron. Yes, chylomicron में वो they will be inserted or they will be packed, we can say. You can see fatty globules combined with the proteins to form a chylomicrons. और ये कहाँ पर हो रहा है? This all takes place, it is inside the Golgi apparatus. Got it, students? Now What are these chylomicrons? These chylomicrons are nothing but they are the proteins which are released at a basolateral surface. या फिर हम बोल सकते हैं कि the chylomicrons they are lipoproteins. Yes. और इनका काम क्या है? This lipoproteins they are generally designed for lipid absorption only. Designed for lipid absorption. Lipid के absorption के लिए these are designed there. So this fatty globules they will combine with a protein, yes, and they will form a chylomicron. अब ये chylomicron क्या होगा? They will be released out, yes, at a basolateral surface by exocytosis. Exocytosis से बाहर निकल जाएंगे from this cell. Now they will be, when they will go out, look, you can see वो blood capillary में नहीं गए, but they will enter the lymphatic capillary or lacteals. We know the lacteals are also present in the center of the villi, है ना? तो lacteals में enter हो जाएंगे. They will not enter the blood capillary. Why they will not enter the blood capillary? Because these are very large to enter a typical capillaries, है ना? Molecules वो बहुत बड़े होते हैं. That's why they cannot enter. So they will enter into the lacteal or the lymphatic capillaries. I hope this is clear. And the micelle, as I told you, which enters inside, once the triglycerides are formed again, this micelle will go out again so that it can help some other fatty acids to enter or to absorb through this brush border of the villus. I hope this is clear now. यहाँ तक समझ में आया? So we can write this in a flow chart. उसको एक simple way में हम लिख सकते हैं. If suppose if it is a short chain fatty acid, yes, then it will be absorbed by simple diffusion, isn't it? Simple diffusion से absorb हो जाएगा. But if it is a long chain fatty acid, है ना, long chain fatty acid जिसको हम या फिर हम बोल सकते हैं उसको मोनो और डाइग्लिसराइड, isn't it? What will they do? They will form micelle. किसके साथ फॉर्म करेंगे? With the bile salts. Yes. Now this micelle internalize enterocyte. Now inside the enterocyte, this mono and diglyceride क्या करेंगे? They will resynthesize into triglycerides. Okay. Now once it is done, they will be packed along with cholesterol yes into chylomicron. Now what is chylomicron? अब मैंने आपको already बता दिया. Yes. Now this chylomicron is released by exocytosis from कहाँ से? Basolateral membrane. And then it will enter into the lacteals, or you can say lymphatic capillaries. I hope this is clear. So this was the absorption of fatty acids, or this was absorption of lipids, we can say. यहाँ तक समझ में आया? So the remaining absorptions are there, the absorptions of vitamins, absorption of calcium, absorption of iron. Ye sari cheezein we will discuss in the next class. I hope these three absorption, that is absorption of monosaccharides, absorption of proteins, and absorption of lipids, which are the three important nutrients. Yes, we have discussed and it is understood to all of you. So we will stop here, students. See you in the next class. Till then, thank you for watching the class, and yes, till we meet, keep learning. All the best for your studies. And yes, you can download the Arth Academy app, because on this app you will get a complete lectures of all the topics for your exams, with detailed notes and many tests also. Okay, I'm sharing the link of the app also. It is Arth Academy app. You can download it from the Play Store, and you can also use this link to download. Okay, so till then, students, all of you, bye bye, take care, see you soon, bye.